resistance is a natural feature of microbial ecosystems. All we've done is given selection pressure for them to amplify and exchange. Was this notion that it's good to question the world, right? And it's good to develop those critical tools that Cody is so amazing at championing.
Good morning, good afternoon, good evening, and welcome to everyone present. My name is Soish from the class of 2023, and I would like to welcome you to Quad Talks 2022, a virtual alumni speaker series where we bring you all together to engage, interact, and be inspired.
Hi, good evening, everyone. My name is Adrian from class of 2022. Tonight's talk is presented to you by KIS and Cody Friends International, an independent nonprofit organization in US that supports the educational mission of the Kodaikanal International School in India and its alumni.
Along with us presenting to you are Alex and Shotaba. Our guest speaker for tonight is Professor Gautam Dantas, a KIS graduate from the year 1996.
Dr. Dantas is a professor of Pathology and Immunology, Biomedical Engineering, and Molecular Microbiology at the Washington University School of Medicine in St. Louis. He received his PhD in biochemistry from the University of Washington and completed his postdoctoral training in microbial genomics from Harvard Medical.
His studies focus on understanding the evolution and exchange of antibiotic resistance amongst diverse microbial communities. His lab develops novel antibiotic and probiotic therapies and engineers microbial catalysts to produce value chemicals such as biofuels.
Gautam and his lab has published many papers, including 8 in Science, Nature, and Cell. They also have 5 patents in biotechnology. Gautam leads an interdisciplinary research and training group with expertise in microbiology, biochemistry, genomics, pathology, infectious diseases, pediatrics, ecology and evolution, systems biology, biomedical engineering, chemical engineering, and computational biology. The lab is situated in St. Louis and is called the Dantas Lab.
He is the co-director of the Computational and Systems Biology Graduate Program. Gautam is a recipient of the AAAS Newcomb Cleveland Prize, the Harvard University Certificate for Distinction in Teaching, the NIH Doctor's New Innovator Award, the Academy of Science, St. Louis Innovator Award, the Washington University Distinguished Educator Award, and the American Academy of Microbiology Fellowship.
Dr. Dantas, it is our pleasure to have you with us today, and we look forward to learning from your talk. But before we proceed, I would like to hand the mic over to my friends, Alex and Shotaba, to go over a quick rapid fire round.
Good morning, sir. My name is Alex, and I'm from the class of 2022. And alongside me is Shotaba, and we have prepared a rapid fire round of questions for you.
Wonderful. Thank you for having me.
Good morning, sir. My name is Shrathava from the class of 2023, and I have prepared the questions alongside Alex. These are mainly questions pertaining to your life at KIS. Please give us the quickest and shortest reply possible.
Yeah, so the first question is, what is the name of the hydrogen?
I'm sorry, Alex, the audio just got out as you were asking that.
Yeah, the name of your dorm.
The name of my dorm was, I'm blanking. I remember the names of the Mr. and Mrs. Sati. Bartlett. Yep.
The second question is, which team or house were you in?
Blue.
Yeah. Third question is, who was your most inspirational teacher or teachers?
That's a great question. I think actually my most inspirational teacher was Bob Granner. He was the English teacher, and I think he put the sort of fear of everything in you at the very beginning, was also one of the most supportive people in the end. So yeah.
What was your favorite place to hang out on the campus?
Probably Flag Green.
Yeah. What was your favorite place outside of campus to hang out?
I think there were a couple of places that you sort of, I don't even really remember the geography terribly well, but that we go hike off close by. So I guess just the outdoors outside of school.
So, sir, were you a hiker?
To some degree. I think back in the day, if you were on the honor roll, you were allowed to leave school at like various points during school, and I would try to, take short hikes away just to experience the outside of Cody, which was awesome.
Were you in any committee like Studco or NHS?
Yeah, I was the president of Studco.
Wow. So, sir, what was your favorite song to dance to at the canteen?
Yeah, that's a great question. I think probably like November Rain, maybe, if I remember that correctly. Guns N' Roses song.
Yeah, and if you don't mind, who were your best friends and their names?
Sure, yeah. I was fortunate to have a number of best friends. There was a couple guys I hung out with, Alan Matamana and Blesson Samuel, with no real hubris. I think we called ourselves the Three Wise Men. It's a regrettable decision in retrospect. But also my roommate, Prithvi Varasinghe, was one of the first people I met and was a very, very good friend to.
So your last question is, what was the one thing your roommate did to annoy you?
Well, I had a number of different roommates, but one of my roommates, my 9th grade roommate, had a tendency to not change his socks very often. And when he did change his socks, had a tendency to kind of handpick stuff out of his toes, which was not the most wonderful thing to be around. So this was not Prithvi, this was someone else. Just
Yeah, okay, so I think we're done with the rapid fire questions, and I think you can proceed to the presentation.
Well, thank you very much. And again, thanks for this wonderful opportunity and the kind introduction and the sort of reminder of these blasts in the past. Embarrassing that I didn't remember the dorm I lived in for 4 years, but came back eventually. Manjushak, should I go ahead?
Yeah.
Okay. Show of thumb to make sure that you can see and hear before I get going. All right, great, awesome. So yeah, it took me a little bit of time to go and pull up some of these pictures. So before I get into my talk, just this was Cody for me many years ago, 26 years ago. This was Alan and Blesson, as I mentioned. This was at graduation. Nina and Buku, a couple other friends. This is actually me back in my sort of John Lennon-y days. Actually, right at Flag Green. And then this is my brother and my parents. Yeah, again, right at graduation. So it's awesome to have this sort of blast from the past. So thank you again for allowing me to tell you a little bit about what my lab and I do. So I want to tell you a little bit today about one of the focus areas, one of the major focus areas of my lab, and that is this interesting understanding and predicting and ultimately combating this scourge of antimicrobial resistance. And I'll hopefully go through and define what these terms mean and what our particular lens at approaching the problem is. But before I do that, I just want to be very clear to emphasize that I'm merely a spokesperson, right? The reason I'm here is because of this incredible group of folks that I have the privilege of working with and representing. We draw people in our lab from a pretty wide background, and obviously I don't have time to highlight every one of their work, but really our lab is us and not me. And furthermore, I think it's really critical for me to emphasize that we not only encourage, but do a pretty active job with trying to recruit diversity across a whole wide swath of backgrounds. We truly believe that you cannot separate us from the science we do. And part of that championing of diverse voices is also realize that as scientists, we have particular responsibilities with the words we use. And I think this is particularly acute given that we're now in finishing the second year of this deadly pandemic. And this is just my little PSA before we get into the science that we have to be very careful about how we label things, right? So especially for those of you who are budding scientists coming out of Cody and will go off to do amazing things, I think, yeah, not only keep your eyes open, but also be willing to challenge old school notions about how people used to label things, right? And in particular, in the field that I'm in, there is this unfortunate legacy of people using people and places and names to define bacteria and the diseases they cause. And I just think that's not a great idea. And it's something that I really hope that we can move towards changing. Okay, so now from that, let's transition to kind of the meat of what I'm going to be telling you about. And that is this recognition that we live on a microbially dominated planet, right? We think of this as a human planet, but it really isn't. If you think of sort of the biological activity, virtually anything that we look at, whether it's above ground or below ground and waters, that the dominant life force are microorganisms, these things that we can't see. And we like as humans, especially those of us who are type A, to kind of label things in terms of particular habitats, but even those habitat boundaries mean nothing to the microbes. And I'm going to use this word microbiomes a lot. And what that really refers to that most microbes thrive in communities, lots of diverse, different microbes in each of these habitats. And we're interested in sort of how they thrive in those communities, how they move back and forth. And in particular, what happens if you challenge those communities with something like an antibiotic? How does that community respond? And so let's go to what is an antibiotic, right? So I'm going to use this word antibiotic or antimicrobial. What they are any chemical that has the ability to kill or block the growth of bacteria or fungi, right? These classes of microorganisms. I'll use the word antibiotic and antimicrobial interchangeably, but really what I mean for the most of my talk, at least, are those specific chemicals that kill bacteria. Again, because we're in the midst of a global pandemic caused by a virus, one thing that they're not, however, are chemicals that do anything to do with viruses, right? So they're not antivirals. And this becomes important because in work that I won't go into a lot of detail, there's a lot of retrospective analysis to show that especially in pediatric populations, so in kids, that oftentimes when kids go in when they're sick and get an antibiotic prescribed to them, it's actually because of a viral infection, and that's not doing any good and actually may do something bad. Okay, now antibiotics were developed primarily for treating infectious bacteria, pathogenic bacteria, and unfortunately, which is the case with almost any evolutionary selection pressure, almost immediately after the deployment of those antibiotics in the clinic, antibiotic resistance arose. That is, the bacteria figured out ways in which to defeat the antibiotics. And if you look at this particular timeline, effectively every important class of antibiotic is put on here in terms of the date that it was originally introduced to the clinic. And where blue shades the red is where the first reports of resistance are against those antibiotics. And the reason this is important is you'll hopefully observe there is no class of antibiotic for which that transition doesn't occur. So another way to interpret this data is to say antibiotic resistance in clinical pathogens is not a matter of if, it's just a matter of when. And that is a key concept to recognize, that we're always going to be vulnerable to those infectious agents if we don't come up with new antibiotics. And that is why understanding resistance and ideally mitigating it is so critical. And that translates directly in an incredible burden in terms of life and money, right? And so there are a variety of reports, and I'll highlight just a couple of them. Drug-resistant bacterial infections currently claim on the order of 1.3 million lives globally. And because of rates of antibiotic resistance and a variety of other factors, the estimate is that that number will balloon by almost tenfold, such that about 10 million people on the planet will die per year by the time we get to 2050, right? Just to put those numbers a little bit in perspective, this number here is greater than the number of people killed by HIV and malaria combined. And by the time it balloons to 10 million, this will be greater than the number of people killed by cancer every year around the globe. So not only is this, most importantly, a human life burden, but it also costs an incredible amount of money. So the rough estimate is that the US economy loses about $55 billion a year due to the treatment of antibiotic resistance infections. And if you take this number and you compute what that means in terms of hit to the global economy will be estimated to lose $100 trillion by 2050 because of drug-resistant infections alone. And I'm just warning you, there is a little bit of a bad news bears component to my talk. I apologize. I'll try to highlight some things, but it's good to kind of understand that the gravity of what we're trying to understand here. And so just to make things a little bit worse, unfortunately, we've had this steady rise in resistant bacteria over the last few decades. at about the same time that new antibiotics are not coming to market. Oftentimes described as the perfect storm, right? You actually would want those two plots to be exactly reversed, but they're not. So this is the situation we find ourselves in. This is the motivation for why we want to understand the impact of antibiotics and ways in which to potentially defeat resistance. Okay, now the problem is actually a little bit more complicated too, because really the perspective I've given you so far is antibiotics against the bad bugs, right? The pathogens, the ones that cause infections. But actually, as I sort of alluded to, microbes are everywhere. They're including in and on us, right? We have more microbial cells in our body than human cells, right? The so-called human microbiome. And as it turns out that those other bugs, those other bacteria are just as vulnerable to antibiotics as the pathogens are. And the reason is, without going into a lot of the sort of molecular details, is because the way antibiotics work, it's almost sort of within their definition, is by targeting the most key processes in the way bacteria keep alive, right? Their cell wall, the way their DNA is replicated, the way proteins are made, all of those key processes which are required for keeping them alive, that's what antibiotics target. And so, not surprisingly, since those are conserved across both the good and the bad bacteria, when antibiotics are deployed, so for instance, if you ingest an antibiotic, you take an antibiotic for, say, a GI infection, you're going to now be interacting with all of those good bugs too. And there's been a lot of work by a lot of other people to show that independent of when you look during the lifespan of humans, right, from birth all the way up until very late life, there are a lot of co-associations between antibiotic use and bad stuff that happens in humans, so-called comorbidities, right? So those are the two motivating factors now. We want to understand how antibiotics work because resistance is a problem with pathogens, but we also want to understand what the collateral damage might be when antibiotics are deployed against the good bugs. Okay, so then let's think a little bit deeper now in terms of how does resistance arise and how do we understand kind of how it will evolve between different bacteria. And so we can turn to understanding how bacteria evolve any function. And the most sort of classically well understood ideas of evolution are what we call vertical transmission. This is how humans evolve, right? Basically by passing on genetic mutations to our progeny. And our bacteria can do the same thing, except their replication rates, their division rates are much faster than humans. And so in vertical transmission, just basically recognize as bacteria go and they kind of divide and multiply, sometime along the way, as those divisions are occurring, mistakes occur, right? DNA gets mutated just by random chance. And every once in a while, that mutation occurs in a spot that changes the function of some protein in that particular microorganism. And if that random mistake occurs in the context of antibiotics, give you a fitness advantage that by mistake it's resistant. Now what's going to happen in that resistant bug is going to take over the population, right? So this is this concept of Darwinian evolution, where random mistakes are made as populations divide, and then if a fitness challenge comes in that kills off the wild type population, the resistant one will fix, right? So this is one way in which resistance arises, but bacteria are remarkable because they can do something that you and I cannot, and they can do this thing called horizontal gene transfer. So there's a vertical, you to your progeny. Horizontal gene transfer, or lateral gene transfer as it sounds, is the ability of microorganisms to exchange DNA, their genetic material, with each other, right? So this would be equivalent to not just you being able to randomly, let's say your friend happened to have the trait of being a really good singer. Horizontal gene transfer would be the ability for their good singing gene to suddenly be transferred over to you and over the course of you having dinner, and now suddenly you're a great singer. We know that that doesn't occur, right? That's not something that we can do. We can't pass traits back and forth genetically. But actually, because of how different bacteria are in terms of their genetic material, it's actually more along the lines of not just you exchanging DNA with your friend, but you exchanging DNA with a squirrel or a banana, right? And again, we know that that's not actually possible, but it's across those large genetic distances that over the course of seconds, bacteria can exchange DNA with each other through this process of horizontal gene transfer. And there are many different processes that I won't go through in terms of how it actually occurs, except to say that a lot of people have established that this method of randomly passing genes back and forth between bacteria is a dominant mechanism by which bacteria become resistant to antibiotics. And if you think about it for a second, that kind of makes sense, right? If you just think of everyone's going to take the shortest route, why would I want to spend all of my time becoming resistant to antibiotics through these random processes when I can just wait for my friend as another bacterium to figure it out, and then I'll just borrow its genes, right? So just in terms, I know I'm anthropomorphizing that a little bit, but that's sort of the intuition of why when you have this high selection pressure, you can have this rapid transmission of antibiotic resistance. And kind of synthesizing the things I've told you so far, the reason this matters is now it means that you can't really distinguish between the good bugs and the bad bugs vis-a-vis antibiotic resistance, right? So say a bad bug came into your body, one that's going to cause a disease, and it so happened that the good bugs in your body already had resistance genes, maybe from prior antibiotic use. Now that bad bug could pick up those resistance genes from your good bugs, and now you'd be hard pressed to treat it. So this is why we need to consider resistance and antibiotics impact not only on the good or bad bugs, but also the good bugs that they could exchange with. Okay, so how do we study resistance? Now, those of you in the crowd who are biologists or scientists of any type have certainly heard of this suffix "ome," right? It's something that we in the field really like adding to old school names. And so you put it the genome or the metagenome or the proteome, the microbiome. It just essentially refers to the collection of all of the particular feature you're looking for. And us not wanting to be left behind also have coined the term the resistome. This is actually something that a colleague, Jerry Wright, coined a while ago. And so when I use the word resistome, I'm referring to the entire collection of genetic material in any group of microbes that could encode antibiotic resistance. So this would be basically, any gene in that community, whether they're good bugs or bad bugs, that could enable the destruction of the resistance against an antibiotic. So how have we studied antibiotic resistance? So one way is sort of traditional microbiology. This is if you Google microbiology and image search, you'll find something like this. And it's basically this concept that in the clinical labs, right, if you or I were to get sick with a bacterial infection, they would go in, they'd take whatever the infectious material was, say a spit sample or a blood sample, and they would culture things up, right? They would domesticate those microbes on a petri dish. And then, so that's the culture. And then they would phenotype them. They would test them for particular properties like antibiotic resistance. That's a great way to figure this out. For the bugs that you care about that you can culture, you can use traditional methods. The problem, though, is most microbes, and we found this especially over the last few decades, are not easy to culture. They're not things that, when you look at even a simple soil sample, there are, tens of thousands of different bacteria in there. And so in this day and age, what you can say, let's skip the culturing step, right, to identify resistance genes, and let's just sequence them, right? This is this concept of you go in and you interrogate what's there based on their genetic identity, right? So, you probably read about this in terms of human genome sequencing. In the COVID age, when you think of your PCR tests, right? What are they doing? They're amplifying genetic material from the virus and then identifying what they are. No different here. Just in this particular case, you're saying, let's say I wanted to investigate what the resistance genes were in a soil sample or in a fecal sample. Rather than going and culturing those bugs up, you just go and sequence all of the DNA that's in there. And the way you'd figure out what a resistance gene was is you'd rely on other people's work. You'd go to a database where other people have described resistance genes and just say, okay, across everything I've sequenced, do I find examples of things that people have sequenced before? And that's why these two methods can iterate on each other, right? People have gone through and very carefully figured out drug-resistant bugs. They've gone and figured out what their resistance genes are. And then you can go and sequence everything else and kind of infer what these are. But because of that, there is a little bit of a conundrum. And that is those two methods are going to be very tightly focused on the known, right? You can't infer what resistance genes are in a new sample without knowing what they were before, right? So if a new resistance gene was to come up, something we'd never seen before, this particular method would fail, right? You can't discover in gene sequence what the function of a gene is if you've never seen it. And so with that method, our lab and many others, and this is a method that's now over 20 years old, use something that is a experimental version, so a lab version of an approximation of that horizontal gene transfer, right? This idea of genes moving back and forth between different bacteria. and this is a bit of a tongue twistery phrase, but it's called functional metagenomic selections. And don't worry about what those words mean. As far as we're concerned today, it's just a complementary technique that is a good facsimile. It's a good approximation of horizontal gene transfer that allows you to say, okay, these are the methods for understanding known resistance genes. This is a method for discovering the unknown, right? It's a discovery tool. So how does that discovery tool work? And again, I'm not going to belabor the details. For those of you who are the sort of biology aficionados, I'm happy to talk about this offline, but the basic concept is you start out with any group of microbes, right? Doesn't matter where they are. They could be cultured or uncultured. You only look at their DNA, and then you force an organism that you know how to work with. So those of you who are microbiologists, you use E. coli, for instance, an easy to work with microorganism in the lab. And what you do is you force E. coli to basically take up little chunks of that original DNA, right? So you're forcing E. coli to effectively go through horizontal gene transfer. It is trying to pick up little fragments of those original microbes and see what they are. And to understand what they are is, you can just turn back to traditional microbiology and take each of these E. coli so-called transformants, right, and put them on petri dishes that have antibiotics in them. The regular E. coli will die because that's the way it was predefined. And the only things that will survive now are those E. coli, those little new bacteria that picked up genetic material from the original source that is an antibiotic resistance gene. And that's your discovery tool, right? That's how you've modeled horizontal gene transfer, and you've been able to show I've discovered new resistance genes because they're genetic material that allowed E. coli to be resistant. And then the devil is in the details. We use sequencing methods to figure out what those genes are. But for purposes of today, really the thing to recognize is this is a novel method that uses both experimental and computational tools to discover new resistance genes in any arbitrary group of microbes as a way in which to basically complement what we already knew before. Okay, all right. So the last intro slide here is just to sort of lay the framework for how we and others try to go about understanding the impact of antibiotics on both good and bad bugs. So we use a sort of, maybe if you think of how clinical trials work, right? You want to understand how a drug works. This happened with the COVID vaccine trials to some degree, for instance. What you really want are cases and controls. And so we use the same thing. Let's say, for instance, and I'm focusing right here, say the cases are all of those individuals who at each of those red dots had an antibiotic treatment. And what you'd like to understand is, how did those antibiotics change the microbes in their gut and the antibiotic resistance genes? So what you do is you get as many cases as possible and enroll as many people who have those perturbations. And then you define a set of controls, people who are as similar to them as possible, same age, same sex, same diet. And then you try to collect samples again all the way through, ideally before, during, and after these perturbations. And then you compare between the cases and controls and within the cases themselves, right? So that's a way in which to infer statistically what the impact of those antibiotics might be. And what are the methods we use? One method we use is sequencing. I've already referred to this, where let's say you're looking at the bacteria, the microbiomes of the gut. You'll get a stool sample, right? A fecal sample. It's a good approximation of what's in the gut. You'll sequence all of the DNA of all of the microbes, the so-called metagenome, the collection of all of the bugs. And then so sequencing methods and basically database comparisons, you can ask who's there, which bacteria, and what they might be doing. You can complement that with the method I told you about before. This functional metagenomics is a way of counting resistance genes. And so now that tells you, here are the bugs that are there, and here are the resistance genes that they might have. And then you can also turn back to traditional microbiology and say, okay, based on all of these hypotheses that I've come up with, I can go back and culture the bugs that I think are most important, and then identify who they are and what they might be doing. And again, there's a lot of details on the slide that you don't need to worry about. Really, the high level concept that you want to take away is, if you want to understand what the result or the correlate of a particular perturbation is, you need cases, you need controls, you need samples both before, during, and after. And then you need methods to interrogate the things that you care about. In this case, we're interrogating the bugs that are there and what they might be doing relative to antibiotic resistance. So what have we done with that? So then the next few slides, I'm basically going to give you sort of high-level views of some of the insights we've gleaned through application of these techniques to understand the sort of landscape of antibiotics and resistance. So before I get into the fancier slides, there's certain images that as a parent freak you out, but also, worth 1,000 words, that resistance spreads across habitats, right? That we don't really have much control over the concept. Well, I guess you have control beyond this questionable parenting moment of the ability to interact with your environment. So, but what this is supposed to capture is this idea that, let's say resistance was enriched in a pig because the pig was fed antibiotics. That might transmit over to you if you happen to be this child. Okay, so this is the more boring version of that particular concept, and that is to say, right, really, you've seen this before. Every single habitat has microbes. You might, for instance, have antibiotic enrichment in the wastewater treatment system, and then eventually that might come back and interact with you depending on where that wastewater went, right? And so I'm going to walk through a couple parts of this cartoon schematic about what we found so far. So I'm going to initially focus on the human microbiome, the bugs that are associated in and on us, right? As I told you, there's microbes on our skin, microbes on in our stool, and I'm going to ask sort of what does that look like in terms of antibiotic resistance. And bear with me because I'm not going to be able to, with most of these things, go into a lot of detail. I'll try to give you the sort of main take-home messages to the application of the techniques that my lab applied that I already introduced. So on one extreme, Erica Pearson was a graduate from our genomics program. We said, okay, let's look at humans who had never seen antibiotics. Now, that's a pretty rare thing. Most of us have interacted with antibiotics, but through a series of really exciting collaborations, we got access to fecal and oral samples from a tribe in the Venezuelan Amazon at first contact, right? And so we were not part of the first contact. There's a group of anthropologists. Samples were collected. As an aside, I leave it as an exercise to you to consider. Imagine you're in the position of these Yanomami Amerindians, and these folks come in helicopters, and the first thing they ask you is for a fecal sample, right? As presumably what first contact with aliens is going to look like with us. But nevertheless, we were not part of this. We fortunately got access to those samples, and then Erica applied the techniques that I mentioned. And again, without going through all the details, what I can tell you is we were kind of surprised to find that in the oral samples and in the fecal samples, the bacteria that were in there, Erica identified resistance genes to antibiotics, even though these people had never taken antibiotics commercially, including antibiotics that were sort of the latest antibiotics. Now, but I'll just give you the sort of spoiler event here. And so why is this? It sort of seems non-intuitive. And the reason really is because antibiotics, the way we think of them, really are natural products. The first people to discover antibiotics did not come up with them in a lab. They discovered that bacteria in the soil, for the most part, produce these interesting compounds that effectively they probably use for their own defense to fight against the other bacteria around them. And all we did as humans is go and discover them. And there are a number of Nobel Prizes that were given out for these discoveries, right? The discovery of penicillin by Fleming and the discovery of the aminoglycosides by Waxman. So many such examples. And so it kind of makes sense in retrospect that resistance is everywhere because those bugs that are in our body and in the bodies of these particular Amerindians had some contact with the bugs that were in the environment. And so selection pressure for resistance is pretty old. But it's still sort of surprising to find this out, right? So, but now think about this. That's our baseline expectation. So based on work like this, resistance is everywhere in these good bugs even. So the ability for these bugs to defeat our antibiotics is already existent. Now, let's look at a different extreme and let's look at preterm infants, right, in the US. So fortunately, with modern medicine getting better and better, we can keep younger and younger kids alive when they're born. Preterm birth is defined as, the definitions change, but essentially a kid that's born that roughly lower than 36 weeks of gestational age. And we can keep 20-weekers, 22-weekers alive now. And for that reason, because these kids are immature in terms of their development, preterm birth is the leading cause of infant death in the world. As you might imagine, like everything else that's immature in these kids, the bugs that are in and on them, especially their gut microbiomes, are not as well developed either, right? They also have that developmental delay. And so here's just an example of looking at the number of bacteria in the guts of preterm infants versus term infants, and they're almost tenfold lower during the first couple months of life, right? So in that context, so they're kind of setting the stage, kids are born preterm, they don't have a lot of bacteria on them. Because of their immature state, they're also highly susceptible to infections. And so what happens? We hit them with antibiotics. And so this is a pretty scary statistic. So this is saying, and these are numbers in the US, but this is true in a lot of other parts of the world, where kids that are born about 10 weeks too early, so kids who are born at about 27 weeks, almost 100% of those kids will get antibiotics during their first couple of days of life, right? So these are just the statistics in terms of the bars that are red are antibiotics. So what you're seeing here is when the kid is in the hospital, when they're preterm, so they usually have to spend a couple first months of their lives in what's called the neonatal intensive care unit. The top 2 drugs that they would ever get in the hospital are antibiotic. 4 of the top 8 drugs are antibiotics. So let's now step back for a second and think of this from an ecological perspective. You're starting with a kid, it's born premature, so it has an immature immune system, an immature microbiome, right? Way fewer bugs to play with. And then you're basically carpet bombing that kid's gut and the rest of its body with antibiotics. And so we would hypothesize that that is going to have some, it's going to engender some problems. It's going to further potentially stunt the development of those good bugs, because those bugs are required for healthy human development. And there's also going to be an insane selection pressure for antibiotic resistance, right? And I say selection pressure because, again, just remember the example before. Those resistance genes and bugs are already there. All we're saying is we're not necessarily inventing new resistance here. We're taking existing resistance and giving it a reason to enrich. And that's sure enough what we found in our first study. And so this was work that was led by Molly Gibson, a computational biology graduate student from my lab, in collaboration with doctors Tarr and Warner, neonatologists and gastroenterologists at our children's hospital here. And here's what Molly did. So Molly was able to access a cohort of samples. So think back now to that case control definition. She had all of these kids were born premature. All of them spent the first couple months of their lives in the neonatal ICU. A group of those kids, our control kids, only had antibiotics at birth, right? So there's no 0 antibiotic kids in this case. But then, they had no more antibiotics after that. And Molly analyzed fecal samples, stool samples from these kids over that period. And then her cases were the ones who had not only those antibiotics early in life, but also at subsequent times. And again, I'm going to gloss over a lot of details just to emphasize what did Molly find that's relevant here. One was that the type of bacteria, and I don't expect you to necessarily recognize these names, but if you were to Google them, and they'll pop up on the WHO website and the CDC website, because these are not the type of bacteria that you expect in a healthy gut. You and I certainly would not want to have these type of bacteria enriched in our guts. These are multi-drug resistant bacteria that can often cause infections. And so that is what we think is one of the consequences of this antibiotic treatment and this kind of hospital environment is these kids' guts early in life at least are getting enriched for drug-resistant organisms. And then the silver lining, however, was that Molly was able to apply what's called a machine learning tool or an artificial intelligence tool. Essentially, those are fancy words for saying, statistical modeling, pattern recognition, where Molly was able to take all of the information from these stools using the methods I talked about, the microbiome and the resistome, and by coding up an algorithm, she was able to say, based on a few features, the presence, absence of particular bacteria and resistance genes, she could, with pretty high accuracy, predict how the microbiome was going to respond. So this was not just an exercising and cataloging what would occur, but ideally in being able to explain what might happen in the future. Right now, this is only 85% accuracy, and our accuracy has improved in the future. But so what is the translation of this, right? These are a lot of sort of jargony details. Why would you care? Why would you or your kids care eventually? The reason is because we've established that there are these consequences, these collateral consequences of using antibiotics, but we're also providing a potential solution for the future, where as these algorithms improve and as sequencing technology improves, you could imagine a future now where, say, a kid was in the hospital 10, 15 years from now, you could do rapid sequencing of their stools, and an algorithm could basically now compute based on that information. Aha, kid X has a particular type of microbiome. We think they're going to respond this particular way to this particular antibiotic. So between our choice of antibiotics that we might have, use antibiotic A for kid X and antibiotic B for kid Y, because those ones are going to cause less collateral damage. So that's kind of the hopeful clinical translation of some of these findings. It's not quite there yet, but that's the hope from something like this. Okay, I'm going to leave humans behind for a second, and I'm going to kind of wrap with taking a dive into the environment, and you'll see why. So I'm actually going to turn to a very different habitat, which I've already alluded to in words at least, and that is the soil. So it's sort of the other end of the spectrum, right? The environment, and we'll be looking at the soil microbiome and the soil resistance. So why look at antibiotic resistance genes in the soil? And I'll tell you three particular papers or findings from other people that really inspired my lab to get into looking at the soil resistance, right? The first was this incredible paper by Jerry Wright's group from now over 10 years ago with a very to-the-point title, Antibiotic Resistance is Ancient. And so what these folks did, which is pretty remarkable, is they went to the Beringian permafrost, this area in Canada that is always under ice. And they were able to core out samples, right? So basically drill into areas in the permafrost that they knew had never been touched by humans. They were able to carbon date those cores to be about 30,000 years old. And then they sequenced the DNA from those carbon core or those carbon dated cores. And they were able to, from that DNA, find examples of resistance genes against modern antibiotics. So very clearly providing genetic evidence that resistance in environmental communities far predates any human use of antibiotics, right? The human use of antibiotics is less than 100 years old. So this is clearly establishing that resistance is an ancient feature. Bugs have had this before we came on the scene. Why? The second part is this thing called the producer hypothesis. Now, this is even older, before I was born. And what the producer hypothesis puts forward is this recognition of what I told you in passing, at least, and that is almost all of these compounds that we call antibiotics are actually natural products of bacteria that dominantly live in the soil, primarily the streptomycetes from the actinomycete phylum. If you close your eyes and you think of a rainy day and you're outside and you think of the smell of the soil, right? That is the smell of the streptomycetes. They're this incredible group of microbes that produce all sorts of amazing compounds, including volatile compounds. That's where you think of that smell of the soil. And the other thing they do is they produce these things that we call antibiotics. And they've been doing it, as far as we can tell, for as long as bacteria have been alive, some millions to billions of years. And so the producer hypothesis puts forward this concept that they did not want to commit suicide, right? They were producing these compounds that could destroy key components of bacterial life. And so they needed to have resistance evolve at exactly the same time that they made production of those. So it's called just-in-time resistance. And so because they've been around for so long, and they can do horizontal gene transfer and they can exchange with the bugs that are around them, it makes sense that the producers, in addition to being the original guys who made the antibiotics, also the original guys who made resistance, and then they basically spread around. And because they're in the soil, then this stuff is perhaps not all that surprising. And then finally, to show that we as humans, as usual, want to have our impact, is to show that in cases where humans do interact with the environment, we provide the selection pressure for those resistance genes to enrich. So this is data that's from Europe over the roughly 70 years or so of, that correlates with antimicrobial use. So antimicrobial use started in roughly the 1940s with penicillin used in the World War. There were a little bit of stuff before then. But what you're seeing here is essentially from these archival soils, these researchers went and counted the amount of resistance genes in those soils over time. And you can see that they steadily increase in correlation with more antibiotic use from humans, okay? So let's put all this together. What does this tell us? It tells us resistance has been there for a long time. We have a pretty good idea of why, and we as humans allow those things to enrich. So the one hypothesis that would come out of that would be to say, aha, okay, fair, case closed. We're fairly convinced if you or I go and get an infection with the bacterium and it's resistant to antibiotic, if I look at the identity of the resistance gene, it should be the same thing that's been found in soils, based on what I've just told you, right? And so the weird thing, though, was when we got to the field, when we started looking at this problem a little over 10 years ago, that little hypothesis that I put forward was not actually true. What do I mean by that? If you looked at the sequences that most people were depositing from the infectious bacteria, the pathogens that cause resistance, and you compared those resistance genes to the ones that people have been found in soil, they were very, very different, right? Their genetic sequences were not identical. Now, that's a little bit weird, right? Because if the things are identical, right, you can assume that if a gene sequence is the same across two bacteria, they probably had either a recent donator or they donated to each other over very recent timescales. Why do you make that inference? Because the further apart two gene sequences are, the more time has probably elapsed for those mutations to occur, right? So we hypothesized that maybe the reason that this evidence didn't exist was not because these hypotheses were wrong, but because the right technology had not been used. That we hypothesized that the resistome in the soil is actually, yes, it's ancient, but it's gigantic. It's really, really big. So we basically had a needle in a haystack problem, right? That there was some such examples out there that we should be able to find these identical resistance genes, but we thought that people just hadn't done the right experiment. And so we thought, let's try doing an experiment that we could test this explicit hypothesis. Could we find resistance genes in the soil that were identical to the things that we'd found in the clinic? And so to do that, we ran a kind of strange experiment. And that experiment actually started when I was a postdoc. And so we went to a series of 11 different US soils. And we dumped those soils into media in the lab that contained antibiotics at exceptionally high concentrations. Now, unless you're an antibiotics aficionado, these numbers are going to mean nothing to you. What I'll tell you is that this number of 1,000 micrograms per mil of an antibiotic is about 50 times for most antibiotics what defines resistance. So basically, anything that would be able to survive in this would have to be insanely resistant, right? In many cases, these concentrations of antibiotics would crystallize in your blood, right? They're very, very high. So initially, we actually did this experiment, just a full disclosure, because we wanted to be a little bit lazy. We're like, that's a lot of experiments to set up, right? 11 different soils, 18 different antibiotics, a lot of replication. Let's just keep these concentrations super high so that most of these things will probably not yield anything. But bacteria are always smarter and more interesting and better chemists than we are. And so this actually yielded a large number of bacteria that didn't really care about these antibiotics, because bacteria in the soil just have these abilities. And in fact, it's something I'm not actually telling you about here, we actually found a lot of bacteria here that actually could eat those antibiotics. Forget about resisting them, they could survive on them. You think of bacteria eating glucose and sugars, forget it. Some of these guys couldn't even eat glucose, but they could eat antibiotics. But we said, okay, that's a weird enough phenomenon. We reported on that. But we also hypothesized that this would be the group of microbes that could be that missing link, that could be that needle in the haystack, because they had such high levels of resistance. And sure enough, Kevin, a graduate from my lab, and Alejandro Reyes, a collaborator, basically applied that functional metagenomics platform I told you, the discovery tool, to look at what the resistance genes were in these particular weird, highly resistant bacteria in the soil. And we were able to now prove this long-standing hypothesis in the field. We were able to find at least 10 such examples now of resistance genes from those particular soil bacteria that could knock out 5 different classes of antibiotics, including every main resistance mechanism. And as an important aside, even though we did this study only in the US, right? We looked at those resistance genes that we had discovered and said, what other people in the world have looked at bacteria that, especially pathogenic bacteria, that have identical genes? And colored in sort of dark gray here are all of the countries where a deposition was made at that point, at least, of pathogens, infectious bacteria that had exactly the same resistant genes that we discovered. So really, supporting our concept that this was basically evidence that was lying in wait, and it was basically a needle in a haystack problem. By applying those new technologies, we're able to prove that resistance is indeed in close exchange between some soil bacteria and human pathogens. Okay, another story that I'm going to tell you about is, so far, really everything I've told you is bad, right? Unfortunately, I've told you sort of, quite often at the end of the talks I give, no matter at what level, people are like, why are we still alive? Which is even more acute in the midst of this pandemic. Well, we're alive because we're actually pretty incredible organisms. We have amazing immune systems. Sometimes that needs boosting. But there is some silver lining here. So why would we do this kind of work? And I'm going to tell you one short vignette about how we can take this kind of scary information and do something good with it, right? And I'm going to focus on one particular class of antibiotics called the tetracyclines, right? You don't need to worry about the details of the tetracyclines, except to know that they're a relatively old class of antibiotics discovered in the 1950s. And they work by basically gunking up the machinery that bacterial cells require for making proteins. Okay, so-called ribosome inhibitors. So because they've been used for such a long time, resistance to them has existed in bacteria also for a very long time. And the two main ways that those particular bacteria resist them is by pumping them out, right? Basically, the drug needs to come inside the bacterial cell to break things. So you can just actively pump it out, right? Or you can basically make special proteins as a bacterium that protects the target. So they have this ability, they call them ribosome protecting proteins. They just make this extra piece that kind of is like a shield. So antibiotic comes in and it can't do anything because it's protected. Well and good, fine. Our lab, and the story I'm going to tell you, through some of these soil surveys, through that method of functional metagenomics, discovered a much more prevalent mechanism of enzymatic inactivation. Now, we were not the first people to show this existed. There's already one known mechanism that did this. And just so, to understand what is different about this is these two mechanisms, right, efflux and protection, do nothing to the chemical that is the antibiotic, right? You're getting rid of the antibiotic or you're blocking it from working. So if you think about it, if you use the sword in the shield analogy, right? The efflux is basically saying if you throw the sword at me, I just pump it out before it hits me. The ribosome protection is the shield. But remember, in both of those cases, nothing happens to the sword. The enzymatic inactivation mechanism is I'm going to break that sword. So basically, you can't hit me anymore, but you can't hit my friends either, right? So that's why it's so critical, because you're degrading the antibiotic. And that's a really powerful mechanism. And so, again, I won't belabor all of the details, but Kevin Forsberg, again, who I had shown you before, now in collaboration with a chemist here at WashU, was able to basically discover 10 more examples of these enzymes in our soil selections. And again, I won't go through all of the gory details to tell you that how we did this, but you'll have to trust me to say, through the work that we did for discovery, we found that these enzymes are much more prevalent than we had previously expected in the soils that Kevin looked at. So now we've discovered a new mechanism of resistance, but at that point, at least, it's not really in the clinic, right? So you might argue now, okay, but Gautam, why are you telling me about this? You've already said that this thing has been around for, since the 1950s. If all of the bad bugs are using these mechanisms, why do you care about this new mechanism if it's in the soil? Okay, well, you care about it because, like any scientist, you hope that your discovery has some impact, while at the same time hoping that, maybe this is one of those times that you hope you don't have impact, because it would mean that something is sort of, good for your science or bad for humanity. And unfortunately, it turned out to be true. That our hypothesis was that we had caught basically sort of the bad actor while it was turning bad, right? That it was a bellwether event. We think that we got lucky and caught this particular emerging mechanism about to explode into the clinic. And unfortunately, and just again, stealing my own thunder, that's exactly what happened. Okay. And so work that Drew in the lab, another graduate from our genomics program, did was to go back to a lot of this work that we had done for screening for resistance across all of these different habitats, and said, okay, Kevin happened to stumble across this in the soil, but are these enzymes now in other places that people just haven't looked at because they didn't have the tools? And at least in our own freezers, Drew discovered 70 more examples of this. Now just think about what that scale is, right? One example in the entire world before this in terms of the enzyme. Kevin discovers 10 more, and Drew discovers 70 more. We don't think our lab is all that good in terms of necessarily doing this. We just think, again, people have missed it because it's just about to happen, right? And sure enough, exactly over the time scale of us beginning to look at this, those enzymes now start popping into the who's who of the bad guy pathogens in the clinic. Okay, so there's our motivation. I don't need to go over all of the details before. We know people are going to die from this if we don't do something about it. And so this is our motivation to dig a little bit deeper. So one more piece of bad news bears news. And so what I'm showing you here is effectively a screen that Drew did. It takes each of those enzymes that came from his stuff from the human gut, as well as Kevin's enzymes that come from the soil. And he tests them in bacteria against all of the antibiotics in the tetracycline class. And hopefully you'll notice that there are these white patches here. So the white patches mean that all of these soil enzymes can only knock out this group of the tetracyclines, but they can't work against this, which is good, right? Unfortunately, these new enzymes that Drew discovers knock them all out, right? That is, if a bacterium now has one of these gut-derived enzymes, they don't really care about the tetracyclines at all. And why is this important? Is because sadly, the tetracyclines are one of the few classes of antibiotics where new things are actually coming to the market, right? 2 of these drugs were only approved in 2018. And so we're kind of excited the FDA has approved them. And lo and behold, we're finding that even before they get into the clinic, there are bugs out there that can destroy them, right? So it's important to know this because it means that as soon as they get deployed, they're basically going to inactivate. So what can we do with that information? we can try to block those enzymes. So this is now an even greater collaboration, bringing in Neeraj Tolia, who's an NIH researcher, and his student JP. And what we did was we said, okay, now let's get a little bit deeper into mechanism, right? And bear with me here for a second. So these are enzymes, right? So the enzymes are proteins that go in and they convert particular substrates into products. In this case, they're converting the antibiotic into an inactive antibiotic. And we want to stop that activity. Why do we want to stop that activity? Because if we block the enzyme, then the antibiotic is still alive, it's not degraded, and it can do its thing, right? So how can we go and block that enzyme? Well, we can try to find another chemical that could basically go and trick the enzyme into getting gunked up, right? Basically feed it a different substrate that kind of looks like our antibiotic, but it's not. And so that way we could potentially give both of those compounds together. One compound goes and destroys the resistance mechanism, and our antibiotic is now still alive. And this idea of combination therapy by inhibitors has been used in the field for a long time. So one of the most important classes of antibiotics, the beta-lactams or the penicillins, have been classically used with beta-lactamase inhibitors. So the beta-lactamases are the enzymes that break the beta-lactam antibiotics. The inhibitors are ones that go and break that resistance mechanism. Used together now, even bugs that would otherwise be resistant to the beta-lactams can no longer be resisted because you break their enzymes, okay? So that's the concept. And we were, I mean, again, glossing over a lot of details. We hypothesized we could do the same thing with the tetracyclines, and that's exactly what we did, right? Now, this is one of those cases where if you were a graduate student listening to this, you'd be a little bit upset at this, is because normally the way this works in science is you have this hypothesis, and you go and an army of people go and spend decades trying to find something of this type. Drew happened to test a compound that we had on the shelf, the very first one, and it worked. Okay, I'm not claiming this would ever work again. We just got lucky that in this particular case, the compound that had the features that Drew was looking for happened to do exactly what we wanted. And so again, you don't have to worry about the sort of details here, except to say that when Drew happened to now mix this particular inhibitor along with tetracycline, we could basically rescue tetracycline activity. So this is the silver lining part of the talk, to say, one of the reasons we go out there and do these discovery exercises is not just to scare the bejesus out of everyone, right? It's to say that if we're ahead of the game, if we have these bellwether events, we can spend the kind of research time required to understand these resistance mechanisms, ideally identify the Achilles heels, right? And in designing then compounds that can destroy those Achilles heels, we can rescue new compounds coming to market. Right. And so to support that hypothesis, again, I'm showing you now just the sort of relationship between the soil enzymes we discovered and the human enzymes we discovered. The closer things are on this particular tree, the more related they happen to be. And unfortunately, as I already told you, we had found, or maybe alluded to this, one of the enzymes we found in the soil we'd actually found in a soil-derived pathogen that causes a Legionella. And regrettably, around the same time that we found one of these enzymes in the human gut, one of the ones that destroys all of the latest generation enzymes, we also found that exact same gene in a bacterium that we've been studying from a long-standing set of collaborations that we have in Pakistan from a patient that was suffering from cystic fibrosis. So why am I showing you this? Is to say the cat is already out of the bag, right? We're discovering it in these sort of benign bugs, but those genes are already in the kind of bugs that are going to cause infections, giving us all of the motivation required to do a better job at finding those inhibitors. Unfortunately, because we work with the Vancevich lab, this organic chemistry lab, we've been able to take that early serendipitous discovery, right, the kind of lucky discovery of the anhydrotetracycline, and then they have done all of this fancy work with synthetic organic chemistry to come up with new generations of inhibitors that we can go and now rescue those latest generation tetracyclines. So I think I kind of told you about this fact that one of the worst things we found was these enzymes in the gut could break even the latest generation drugs. But because that was just happening and we had this little bit of time ahead of us, we're able to discover these new inhibitors and rescue them. So this is a bit of a story to say, yes, there are scary resistance genes out there, but as long as we use appropriate methods to discover them before they're a problem, we could hopefully provide solutions, right? Solutions that could actually help keep our current therapies alive. Okay, so the last thing I'll tell you about, which is about 2 or 3 slides, unfortunately takes you a little bit more into the deeper and darker territory. This is not the good news bears part of the presentation. Just to say, I've talked about individual habitats. I've talked about humans on their own, and talked about soils on their own, and the resistance genes that are there. But we live in an interconnected planet, right? So what if we were to consider a whole bunch of habitats at the same time? What about the transmission dynamics of humans and their environment and their animals? And so the one example I'll give you here is a study that we conducted here to test exactly that. And this was a huge study. It was basically two students' PhD thesis, Pablo Sukayama and Erica Persson from the Micro and Genomics programs in our lab. And they independently ran these long-term studies in our lab in collaboration with a bunch of people local in El Salvador, where we looked at a group of individuals living in a village, and in Peru in a peri-urban slum. And the reason we looked at these two particular habitats with longitudinal sampling over this has happened about 10 to 8 years ago, is we wanted to go into high density human environments that represent a much more reasonable part or better representations, we thought, of the world population than all of these studies that occur in, for instance, US populations, right? We're just as guilty being in the US as looking at sort of Western centric questions, but most people on the planet live in either agrarian or semi-urban or peri-urban settings. So we wanted to ask, in these environments where people are interacting a lot more with their environment, what does the microbiome look like over time, and what do their resistance genes look like? And our main goal here was to understand transmission dynamics of the bugs. Now I'm going to just completely gloss over about most of what we found and tell you that through this work, applying the same techniques I told you about, one of the greatest deliverables was, from a public health perspective, we were able to identify hotspots in both of these locations where it appeared that resistance transfer was occurring between habitats, right? And in this particular case, in El Salvador, it appeared to be the chicken coops. So these folks keep chickens as subsistence chickens, so that they use them for their eggs and for their meat. And then in Lima, in Peru, we identified their wastewater treatment as the two hotspots for where resistance gene exchange was occurring. How did we do that? Again, without belaboring all of the details of the methods, we're able to say, okay, the resistance genes that we find in the chicken droppings, right, are identical to the resistance genes we're finding in the humans and the soil around them. So that's why we hypothesized that's the connection point. And because this study was longitudinal and we did a lot of GPS-based tracking, we're able to find that these chickens, because these folks live in huts, those chickens are in their coops, but they sometimes also just walk into the hut where people are cooking and they walk out. And so that's what we think the transmission dynamics are, right? It's literally the physical interactions. And so that's how we think the resistance is moving around. Here it's a little bit different. And this is something that might be more relevant to a larger swath of the population. So what's happening here, right? What is happening in this wastewater treatment plant? It's probably like a wastewater treatment plant that all of us have access to in some capacity. All of the waste from the Peruvian system, in this case in Lima, comes in. There's some excellent civil engineering to kind of degrade microbes and remove them. That's what it's kind of set up to do. But it doesn't do anything to DNA, right? That's not, those civil engineering techniques were never designed to do anything in terms of the genetic material. And so what we think happens is the bugs get killed off, but their DNA floats around. And there are some bugs that basically survive in these particular conditions, environmental bugs, and they, we think, are picking up those resistance genes that are coming in from the waste stream, which is basically all of the bugs that are in our guts, right? But why is this a hotspot? Is because Lima is a desert, okay? There's not a lot of water around. So what you're seeing here in this picture are trucks that go around Lima to irrigate all of their parks and fields from the so-called gray water, the thing that's been cleaned up in this particular system. So now think about what I mean in terms of a hotspot for resistance dissemination. This is the stuff coming in. Bugs are being killed, but the resistance genes are surviving. Those are being picked up by the environmental microbes. And then the so-called clean water is being sprayed all over the place where you and your kids are gonna go and play around in those particular parks and perhaps pick those genes up. And so that's the reason why we think this is something that needs to be paid attention to. Now, we didn't do any of that, additional public health work. I just wanted to end with this to show you that this is another application of basic science, right? Basic research intended to discover microbes and resistance genes, but it could be potentially used for impacting global public health policy. Okay, so with that, I'm going to end and say, this is what we look like sometimes. This is what we look like at other times, right? These are lab, in the more sort of staid formal settings, which we don't actually do very often. This is us. In Missouri, we do this weird thing called float trips, where you hop into rafts and just float down a river for a day to hang out with people. And our lab likes doing that as a morale building exercise every once in a while. Harder to do in the pandemic, but hopefully we'll get to be around that hump. So many different people to thank. I just don't have the time to do it, except to say, just sort of in general, everything of interest that I've said to you is because of the incredible people that I get to work with, as well as many alumni have gone on to bigger and better things. We have incredible collaborators, both locally within WashU, as well as around the country, as well as around the world. I talked about some of them in El Salvador and Peru and Pakistan. We've been very fortunate to be supported by a number of federal and private agencies. And I'm happy to answer any questions, but leave you with this particular PSA that, certainly take the antibiotics that are prescribed to you if they're for a bacterial infection, but please do not use antimicrobials for viral infections. They will do no good, and they will almost certainly, as you've seen, do some harm. So I'll end there, and happy to chat.
Thank you so much, sir. On behalf of everyone present today, we would like to express our gratitude to you for your extremely informative presentation. We sincerely appreciate you taking time out of your busy schedule to hold this session today. Now we will move on to the Q&A session. I request everyone to not hesitate and to ask any questions that you may have.
All right, so what we'll do is we'll change everyone to panelists so that we can have a more interactive session. But I think there are a few questions in the Q&A box. We can start with that. I think the first one is, why do you call yourself the three wise men? It's a very pressing question. Hello.
It is. It was, I think it was the, teenage hubris is probably the easiest way to explain that. I don't know. We've just loved sort of hanging out together and, fancied ourselves as somehow deserving of a moniker, and that's the one that stuck. So that's what we call ourselves, the three wise men.
The next one is, how is it possible that resistant genes to modern antibiotics were present in people who had never taken antibiotics?
Yeah, wonderful question. And so that is now trying to put together a few of the different pieces of evidence that I put forward. So first of all is this recognition that antibiotic resistance, as I showed in the soil, is ancient, right? Other people have demonstrated this. And you understand that microbes interact across habitats. So that means that even though the bugs that are in us right now have been around in human habitats for a long time, they probably exchanged and interacted with microbes in the environment. And so because the microbes in the environment have had resistance genes for such a long time, that's essentially the explanation that we proffer right now as to why would there be resistance to modern antibiotics? Because even this concept of a modern antibiotic is a bit of a misnomer, right? We discovered those antibiotics, or maybe synthetically modified them over shorter timescales over the last 100 years. But those compounds have been around for a gazillion years, right? For a very long time. Resistance mechanisms have been there for a really long time. And so this is really sort of emphasizing this idea that resistance is a natural feature of microbial ecosystems. All we've done is given selection pressure for them to amplify and exchange. So that's the reason why we think that these people who had never seen antibiotics were kind of pre-deployed. Another way to think about it, just from a microbial perspective, is the microbes have had all sorts of reasons to defend themselves against attack. And so they come in pre-armed, and then when we deploy antibiotics, they defend themselves.
I think, Alex, you had a question?
Yeah, I'll go ahead and give a question, which is really a subject-specific question. And if I'm not mistaken, the imminent threat the world faces pertaining to microbiology is antibiotic resistance. And so my question is, are there substitutes for antibiotics, and how effective are they? I've heard about phage therapy, and yeah.
Yeah, excellent question. And so It's, and our lab has some work on this, just don't have time to go through this today. So yeah, so, I guess the basis of your question is, if there are all these dangers with antibiotics and we can't come up with new ones, what else can we do, right? And so there are absolutely alternatives being considered. You talked about one of them, but I'll put that under the global umbrella of, I think, to philosophize a little bit, I think we've been using the wrong language in terms of thinking about antibiotics. We've been using the language of warfare, right? We think of them as enemies and we want to destroy them. And the problem is, when you do that, you give them amazing selection pressure to want to survive, right? You want to wipe them out and they want to survive. And so if you think about how microbes actually interact in the natural world, they live in communities. And so the argument here is, if we were to understand the language of diplomacy to some degree, right, we might have an opportunity from those particular interactions to use alternative strategies to go after the part of the microbes that we want to, and that is, we don't want to kill them necessarily. We just want to prevent them from killing us. We want to stop their pathogenesis. We want to stop their virulence, not necessarily to wipe out the bug. So what are ways in which you can do that? So one way is to actually develop narrow spectrum therapies. By narrow spectrum, I mean they're very targeted, very precise against the type of things that are actually killing us. And so if you think about, what are ways in which bacteria cause problems for us, oftentimes they produce specialized proteins which are toxic, and they'll go and punch holes in our blood cells, or they'll cause issues. So, well, I don't care about that bacterium, I only care about that toxin. So there are a number of strategies that people are coming up with, which are very targeted against annihilating the virulence factors, those pathogen factors. So that's one, right? The other is, as you already alluded to, look at natural mechanisms by which bacterial populations are kept in check. So I touted to some degree that we live on this microbial planet, they're the dominant life forms. That's a bit of a misnomer, right? So the most dominant DNA containing or nucleic acid containing organisms on the planet are actually viruses. There are more phages in the planet than there are stars in the universe, right? And what do those phages do? They're the natural predators of the bacteria. So because they do that already, there's a lot to learn from those interactions to say those predator-prey relationships are ones that we could maybe through biotechnology harness, right? But again, we don't need to necessarily harness them from a pure warfare standpoint. Those phages also modulate particular parts of bacteria. They stop them from making particular things. So that's what I think where the therapies of the futures will come, will be much more narrow spectrum, precise, tunable therapeutics that learn from the biological world. And so stuff that our lab is doing, for instance, is we're trying to generate engineered probiotic organisms, bugs that are good, right? That we would like to be able to engineer specific, proteins into that'll deliver payloads, biological payloads, very specifically to parts of the body, and then use them as alternative to antibiotics. Now, none of that is ready for immediate prime time, but we're getting closer, right? So we have some exciting work, for instance, in our lab, looking at one of the main threats, at least in the US, and a lot of developing nations also, is this bug called C. diff, or Clostridioides difficile. It causes a lot of death and illness, and we have some probiotics right now which might be able to make targeted sort of precision anti-C. diff therapeutics that you could ingest. Basically, you can ingest this thing that'll go in, modulate C. diff, and actually work as a sponge attached to C. diff and take it out without killing it, and that could be a potential therapy for the future.
Hello, Dr. Gautam. Sorry. Yeah.
Go ahead.
Yeah. Have you come across any examples wherein the beneficial species have turned to be pathogenic due to mutation?
Wonderful question. And so this is something that again comes down to, we as humans like to classify things, and it's just easier if we think of things as black and white, good and bad. And that's just not the case. It's actually a spectrum. And honestly, microbes don't really think in those terms, right? They're not sentient necessarily from the way we think about this. And then they're not necessarily structured. Think of like, oh, I'm going to do something good to a human or bad to human. They're opportunists. And so there is this concept of a pathobiont, I think, that is catching, that's getting a lot more attention, to this idea that even bad bugs in many cases are context dependent, right? So you have microbes that interact in and on us, and then depending on what context they are, they might help us, they might ignore us, or they might do bad things, right? And so even the example I just gave you, C. diff, So this is a microbe that a lot of us can carry in our guts asymptomatically, that it does nothing. In fact, it exists as a spore. It is not even sort of living and thriving. But unfortunately, if you were to take antibiotics for other reasons, one of the things that happens with those antibiotics is you suppress a lot of the good bugs in the human microbiome. And those good bugs, one of the things that they're doing is they're playing playing defense against C. diff. They're keeping it dormant. Once those competitors are gone, a lot more resources are available, a lot of food is available for C. diff, and so C. diff wakes up. It gets out of the spore state. And sometimes when it does that, it starts producing this toxin, which ends up then being pathogenic to us. And so that's a great example of a bug that in one state completely benign, but because you wrecked the microbiome by using antibiotic, it basically wakes up and then can cause issues. So that's one example of what you were alluding to, where so-called good bugs can go bad in a context-dependent fashion.
Thank you. Yeah, I have another question. Will vaccines against bacterial diseases be an alternative for antibiotics?
100%. Yeah, that's a great idea. And there are actually already vaccines against bacteria that we have. So, some of the pediatric vaccines we take, even though many of them are viral, some of them are bacterial vaccines. So it's, in fact, ideally all of us have already taken bacterial vaccines for some of the deadliest bacteria out there. And there are other vaccines in development too. And I think the mRNA technology that's come through and made such a big splash with dealing with parts of COVID, I think will be huge, because one of the issues with vaccine development is, at least traditionally, it's taken a long time to basically run through the process of taking it to market. That technology has been a game changer, so I expect that we'll see more and more vaccines. And what's neat is that even if you were not to come up with a vaccine purely, you can use the concept of what vaccines do to help with next generation therapeutics. So I think the question that Alex posed about alternative therapies. So one of the things that we're doing with probiotic engineering is we are engineering microbes to display on their surfaces antibody-like molecules. So we're taking the vaccine concept, but marrying it to probiotics, and to say, okay, now what is a vaccine in many cases, right? It's a way in which to make your immune system a little bit better at recognizing the pathogen and taking it out, right? So what if we were to trick out our probiotics to become next generation vaccines? So it's something that we're working with a couple of biotech companies here to basically engineer our probiotics to have those particular molecules on the outside, so they can work as sort of either vaccine adjuvants or vaccine alternatives to be able to do something very similar. But yeah, absolutely. I think that's one part of this alternative to antimicrobial strategy.
Thank you.
Yeah, Shrotiba, go ahead.
Yes, so sir, coming from someone who is themselves resistant to antibiotics, according to you, what is the single most significant factor driving increasing rates of antibiotic resistance, and what does this mean for medicine in future?
Yeah, great question. So I think nature abhors a vacuum, and so the reason we have antimicrobial resistance is because we use antimicrobials, right? It's unfortunately a pretty simple equation. The more we use those particular compounds, the more we're going to provide selection pressure for resistance to occur. Facetiously, we often talk, within our group and within the field that, antibiotic resistance is like death and taxes. It's inevitable. It's always going to happen. There's no concept that we can have that resistance won't arise, and it's because we have to use antimicrobials. And so, what can we do about that? Well, one of the things we can do is just use our antibiotics better, right? We have to be better stewards of this natural resource, like anything else, right? Like our water and our air. Antibiotics are things that we were given from the environment, and we've been abusing them, right? We use them wantonly, and so that's one part. We just need to be better at it. We need to, as parents, be a little bit more judicious to not demand antibiotics every time our kid has the sniffles, because it's probably not useful, right? The other is we need to spur development of new drugs, and that might be new antibiotics, but also, like the other questions that have come up, with novel therapeutic options. And then I think just sort of, and it almost sounds like when I say basic common sense, I think we just have to look at the state that our world is in to say that that's in short supply. We just have to also be better at understanding how infectious disease works, right? We need to trust our systems when they give us advice about public health issues and say, okay, right, if we're being told to wash our hands and to mask up, we should do that, because that's going to have another layer of defense against what the infection might be. Now, when I say mask up, that's not going to do anything for a bacterial infection. I just mean we need to pay attention to the public health professionals that tell us how to keep ourselves safe.
Manjusa, shall I ask one question?
Sure, Prem, go ahead.
Yeah, thanks for this informative talk. I want to know, how close are we to running out of chemotherapy?
How close are we to running out of antimicrobials, are you asking?
Yeah, that's the thing.
How depressed do you want to be this evening? I would say many of us in the field don't talk about what happens if we get to a post-antibiotic era. I think we've already resolved ourselves to think of ourselves that we are in the post-antibiotic era, right? We have run out of our antimicrobials for all intents and purposes. The latest generation drugs that we have access to that are truly novel were discovered at least 20 years ago. We don't have new drugs coming to market. So from that perspective, right, if you look at the rates of infection and the rates of drug resistance, they are worse than pretty much any other global threat on the planet, right? The reason that antibiotics and antibiotic resistant infections don't get as much press is because, unlike COVID, for instance, they don't kill us quickly. They're a long-term kind of distributed threat. And so, yeah, so, again, sorry to be the bearer of that bad news, but we have to be incentivized to come up with new therapeutic strategies against infectious agents because the bacteria are not getting any less drug resistant. Right? And you just have to look at reports of, there are now bacteria, as an example, that have been found that have ultimately killed the people that they infected. They're resistant against every single antibiotic we have in our arsenal, every one, right? Bugs that have 50 antibiotic resistance genes in their genomes. Kind of selfish, right? But so yeah, the answer is we have run out as far as for all intents and purposes.
Thanks, Gautam.
Go ahead, Zoish.
Okay, so I had a question related to your past, basically. So how did you come up with your field, like after school? Was it like all planned in high school, choosing subjects, or did it was last minute?
Yeah, it's a great question. And I'll try to do this somewhat briefly, but a huge inflection point was Cody, right? And I think the Rod Smith that's on here is the same Rod Smith. But when I was in Cody, it was a transformative experience for me. I was a standard nerdy kid in Bombay, didn't do anything besides school. And then, Cody was eye-opening. Manjusha's dad was a huge transformative force in biology. But Rod, for instance, right, he came in and kind of pinch hit one semester. And he was a biology teacher. And he kind of, I think the very first day, talked about this almost National Geographic scale type of experience, right? Talked about being a marine biologist and doing all of this amazing stuff. And I think he just name dropped, at least in my memory, the phrase biochemist. And knowing absolutely nothing about what that meant, I was like, that's what I want to do. It sounds super cool. And honestly, maybe it was just because that's my personality. I was like, until I figure it out, I'm going to go get a PhD in biochemistry. Not a great plan because, right, but I think why I said not a great plan, it was not because I had this idea that I would be here now the way I am. It was just because Cody, for so many people, was just so inspirational that I was like, this is what, this has caught my fancy. And so I went, with the assistance of the guidance counselor at the time, found a liberal arts college that had a deep history in terms of supporting international students, but also having a science curriculum. I went and got a biology and chemistry degree there, and used that as a stepping stone to go and get a PhD in biochemistry. And interestingly, the reason I'm giving you this history is what I eventually did for my PhD in biochemistry had nothing to do with the work that I did with Mr. Nainan at Cody, or even at the research I did at Macalester. I did, my PhD was in computational biochemistry. I did structural biology. I solved structures using X-ray crystallography, which was awesome. It was fun. But it was mostly because I think those training exercises are about learning to be someone who questions things, right? And that's that's the bug that I caught when I was in Cody. And then based on that inspiration, after I finished my PhD, I was interested in doing something more applied. So I decided to go in microbiology. And then the stuff that we do now, again, is highly unrelated to what we did then, because I think the relationship was this notion that it's good to question the world, right? And it's good to develop those critical tools that Cody is so amazing at championing, right? And so, yeah, so I know, long-winded answer to say that's the reason.
I'm going to add to that. There's a question in the Q&A box asking, do you have any tips for students who are looking to study biotechnology in university or biology in general?
Yeah, so I'll try to give you two answers to this. The most practical part of it is do what you find keeps you up at night, right? That's not I mean, I suppose that's not even really a practical answer. It's the more philosophical answer. What is it that is going to make you feel excited? Don't do what your parents think would be a fun thing for you to do, right? Because if you're not jazzed about it, right, especially with science, I tell my grad students all the time, science is a lot more about failure than it is about success, right? It's about trying things and be happy about the fact you're like, nope, 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 nope. And then every once in a while there's a yes. And oftentimes that yes, you probably even shouldn't take credit for because it was a serendipitous yes, right? It's to be in the weeds of those no's and understanding why those no's are exciting is what makes you a scientist who will survive the system, right? And so now, what would you do in terms of practically getting into biotechnology and biology? Well, first of all, ask yourself, do you care about biotechnology or biology? Do you care about how the world works on the living scale? And if you do, then go do that. And then the kind of coursework that you might want to get involved in, especially with biology now, is I strongly encourage you to take a spectrum of experiences with, you know, the biological sciences, but also the quantitative sciences, because that's really where I think it's the interface of those disciplines that I think will be most useful to you. So if you want to do biotechnology, right, take courses in computer science and take you know, immersion in informatics, right? I think right now the most important thing that you can do if you're a high school student in Cody is, you know, take the diversity of courses that is going to excite you.
Thank you. There was another question. What does the word OME represent? Is it also related to chromosome?
Yeah, well, I mean, those of us with connections to India, Om, right, it represents to be one with the world. But in this particular case, it's a sort of systems biology concept. It really says whatever you want to study, right? The Om addition to it recognizes that you're looking at not at the individual level, but at the systems level. So genome, as an example, is each of us has genes that are part of our DNA makeup, and that entire DNA makeup in the human body is the genome, or in the bacterial cell is the genome. And so really that's what the Om suffix really is to refer to whatever that prefix is, whatever it's being added to. It's the collection of all of those entities in the particular sort of frame that you're studying. So that's why when you get even you know, greater, which is why concepts like metagenome exist, because it's the genome of all organisms now in a particular community. So all of the bugs in a community, all of their genomes together, the metagenome.
Thank you. Is there any questions from the group? I'll continue with the Q&A otherwise. Right. All right. There is a question that says, what is the current state of development backup, emergency use, the last line antibiotics? Are we in a good place with available stocks?
It's a slightly complicated question in terms of, I don't know what the actual stockpiles are, right? I think a lot of drug development, especially the production, occurs in India and in China. So I'm sure that because of the status of you know, the pandemic with supply chain issues, there are shortages ongoing. But really the bigger question is, even if we had massive stockpiles of so-called last generation drugs, if you've got bugs that are resistant to those last generation bugs, it doesn't really matter if we have big stockpiles of them. So at least as of now, on local timescales, right, over the course of like a couple years, I don't think we're actually running out physically of the drugs themselves, right? Those are probably available. Now, of course, I say that from a position of privilege. I live in the US, right? And so I have access to those drugs. Certainly, there are critical parts of the world that don't have access to those drugs, exacerbated by poverty, exacerbated by the inequalities that of COVID. But outside of that, we certainly have, if we were more equitable in our distribution, there are drugs available. The problem is those drugs are losing efficacy. Right. And so that is what we're running out of. And that's what we need new drugs to, the motivation for why we need new drugs.
Thank you. The next one is from Clarence Maloney. Actually, there are two questions from him, so I'll read them together. The first question is, a KIS student tested Cody Lake water and found heavy microbiotic resistance. Another found heavy anti-fungi resistance. What can be done about it?
Excellent question. So, I think, first of all, we shouldn't be surprised that environmental microbes have resistance to these compounds, kind of what I talked about earlier. The question is, what are the levels of those resistance genes? What bacteria are they in? And what is the likelihood that a human would come into contact with those particular microbes? So there are many, many layers of kind of assessing the threat of those resistance genes. Just finding resistance genes in these various places is something that we should expect now, right? Partly because, as I said, this point is ancient. Where things get a bit hairy is if, for instance, those resistance genes are found in the type of bacteria that can cause diseases in humans. Then that's a much bigger threat, because now if you fall into Cody Lake, right, then it could be a problem because you could get infected with that bacterium. So I think to contextualize that finding, we need to understand, what are those particular bugs that are carrying those resistance genes. And then what can you do about it? Unfortunately, I don't think we have the technologies to mitigate environmental level resistance at this stage, right? Cody Lake is pretty large, right? It would be a futile exercise for us to try to go and say, we're going to try to inactivate antibiotic resistance genes everywhere. I think you can make longer term public health interventions that could help, not with that local problem, but just, sort of things in general. And those go back to the kind of points that I made, right? Like, we need to be better with our antibiotic use. A big elephant in the room that I didn't refer to because I didn't present work on it is that, at least in the US and in a number of other countries, I don't quite know what the situation is in India, but a lot of antimicrobials are actually used as growth promoters for food animals. Right. So in fact, globally, by weight at least, more antibiotics are used to basically make our meat cheaper for those who are non-vegetarian than they are used to treat humans. And the exact mechanisms for those are not perfectly well understood. It might be to reduce infections. It might just be because it helps with growth promoting in another way. But it's a terrible practice, right? I would argue that's a huge waste of that natural resource, because not only should we not worry about making our meat cheaper, there are other alternatives. And I'm a non-vegetarian. I'm not proselytizing there. I'm just saying there's better uses of our antimicrobials, but also just the recognition that we're running. If you think about it for a second, this is kind of terrible, but the biggest evolutionary experiment that humans have run in history is the use of antibiotics in food animals, right? Because what have we done? We've given all of the selection pressure we want for the animals and their microbes to basically become the microbes in those animals to become resistant, and then we consume those animals, right? And we consume the microbes that come with them. So again, that's not a direct. I don't think we're raising food animals in Cody Lake. Hopefully, you're not, right? But I think in terms of what we can use with that information about environmental resistance is we can just be better stewards of that natural resource.
Adrian, would you like to go ahead and ask your question?
Yeah, I had a doubt about a point that you made in your presentation, where antibiotics affect all bacteria. It's not like we can target it towards one species, right? So why do some bacteria, such as streptomyces, produce antibiotics which could potentially harm themselves?
Yeah, a great question. So a few nuanced clarifications. So it's true that antibiotics tend to be broad spectrum, but sort of the details, that's not entirely true. There are some narrower spectrum antibiotics. So some antibiotics work against only gram-positive bacteria versus gram-negatives, but the general point remains that they're not only targeted at drug-resistant, at pathogens. In terms of your streptomyces question, right, why is it that they produce compounds that could kill them? It's a little bit hard to ask the why do they do things, because we can't communicate directly with the microbes. But what we know is the way they solved that problem some way in a way was to make sure that they had resistance at the same time as producing those compounds, right? And there are a variety of ways in which they could do that. So if you look at the genomes of producer organisms, you find that they quite often have efflux pumps in them, right, to get rid of the compound. So basically, as soon as they make it, right, just in time they get rid of the compound. So effectively, they can't kill themselves. Or they'll produce those ribosome protection proteins. Now, if you go back even a little bit further in terms of thinking, but still, why make those particular compounds, is where things get a little bit dicier, is that we call these compounds antimicrobials because when we have discovered them and produced them in high concentration, we can kill pathogens with them. However, those compounds are never produced at those same concentrations by the original producers. They produce at much lower concentrations. And so there is the possibility that those compounds were never actually even made as offense molecules. That naturally, the role of antibiotics may not be as antibiotics. They might be communication molecules, right? And there's a good theory to say that that's exactly what they are. And why communication molecules? Well, let's think for a second. Let's say you wanted to send a message to your friend, right? But you wanted that to be a coded message. You wanted it to be an encrypted message, a message that no one else could hear. Wouldn't it be really useful if that message would be toxic for other people to get, right? So maybe that's why, right? Like it's kind of a happy accident that this is something that the streptomycetes are using to communicate with other streptomycetes to be like, hey, I'm here, I've got food, come and grab me, right? And some other bug comes in and goes, okay, I'm going to steal that message. Well, not so fast because it's going to kill you, right? So again, this is a bit of speculation, but that's why. So there's one answer is they have just-in-time regulation of their own resistance mechanisms, and B, it may not even be an offense molecule in the first place.
Thank you. Ernie, would you like to ask your question?
Yeah, thank you. I'm a family physician in Canada, and I suppose maybe I'm one of the boots on the ground. I see a fairly bright picture with antibiotics. I do take seriously the doom and gloom that we have with antibiotic resistance and how we need to be careful about that. But I do see a lot of patients with infections, and I don't see the antibiotic resistance that often. I would say I haven't ever seen a case in my practice of a person with a superbug that died of it. I do see some MRSA and vancomycin resistance. I guess, are we in the first world seeing still good outcomes with antibiotics, or is the third world in other countries, is this a much more common thing with these deadly antibiotic resistance, or could you comment on that?
Yeah, a great question. So I think an important part to nuance the doom and gloom that I was presenting is the problem is dire, but it's not dire in the course of days and weeks. It's dire on the time scale of years and perhaps decades. And so that curve of drug-resistant infections is shallow, right? And so all we have to do is look at drug resistance rates, even in the US or Canada now versus 20 years ago, right? And the levels of resistance that are seen kind of, at least on an average pattern, are higher. So yes, it's true that right now antibiotics are still quite effective, that many of the bugs that we're trying to treat are susceptible to at least some combination of drugs. We also have stepwise programs if you're resistant to this, right? So the bacteria haven't accumulated every resistance gene out there. And there are also reasons for that. If you think about the actual resistance mechanisms at play, it is costly for bacteria to necessarily maintain all of those resistance mechanisms. So if there isn't selection pressure, right, if there's not constant antibiotic use, those bacteria will sometimes shed those antibiotic resistance mechanisms. So there's this concept, even as a physician, you're probably familiar with this, is almost this sort of crop rotation idea, right? That is, you use one antibiotic for a while, and then you stop using it to relieve the selection pressure and move on to another antibiotic, right? So the same way that you keep your fields healthy by rotating, different types of crops, and you change your resource utilization, you could do the same thing with antibiotics. So there are stopgap measures in terms of basically extending the lifetime of our antibiotics. We're not going to die from drug-resistant infections right now. The problem is we don't have new antimicrobials coming to market, and the levels of resistance are steadily increasing. You're right that in areas of higher infectious disease burden, areas of greater inequity, drug resistance issues are higher, but we are not immune to that here in the US or in Canada. It'll eventually catch up with it. And I think the reason that the sort of alarm bells are being rung by those of us who work on this is to say, antibiotic resistance and drug infections is the kind of problem where if we are reactionary about it, if we wait until it's a problem, it'll be too late at that point. We won't be able to do anything about it. So we need to activate now to basically ward off that cliff that we're, essentially falling off of.
Thank you.
Okay, Gautam, I'm going to take something from the Q&A box, and we have another 10, 15 minutes before we wrap up. Glenn Rogers asks, what are other notable hotspots, such as your Lima example? Is the Nile Valley another hotspot?
Yeah, it's a great question.
I just wanted to clarify, help us understand the geography and what are the implications of, is there a network of hot spots, and is there something, whatever the opposite is, a network of cold spots?
Yeah, Glenn, great question. Yeah, so when I was talking about hotspots there, I should clarify, I was not necessarily advocating that El Salvador or Lima in particular as geographies are hotspots. It was in those particular geographies we were able to identify across the various ecosystems that we had, or habitats that we had surveyed, which were the more likely sort of hotspot points for transmission, right? So again, I didn't have too much time to talk about, and maybe I can elaborate, like both in the El Salvador and the Peru example, we got fecal samples from humans as well as the animals that were in and around their households, whether they're subsistence or pets. We surveyed the soils, we surveyed the water, and in this case, the sewage system. So we were looking at all of these different interconnected environments. And then for each of those environments, we were asking the question, which ones are most likely to be the conduits of resistance between the environment and humans? So in El Salvador, in that particular village, we were able to identify the chicken coop as the hotspot for that local environment. For that particular peri-urban slum in Lima, we identified the wastewater treatment system as a potential hotspot because of the particular vagaries of, in a lot of cases, if you look at your local wastewater treatment system, the waste is probably, dumped somewhere else. This just happens to be this unique situation where that gray water is recycled, right, and comes back into sort of interaction with humans because of its reuse, and that could pose a particular problem. So, to answer your question, what other hotspots or perhaps cold spots exist? I don't think, at least none of our work is implicating particular countries or particular locations per se as being the dissemination points where lots of resistance genes are going out. It's more the idea that resistance is everywhere. We as humans are vulnerable in a sense to our environmental interactions. And for each local geography, because of those interactions, we can use tools of this type to understand of all of that resistance out there, which are the ones that are critical for jumping over, and then we can mitigate, right? And so I do think others have also shown that wastewater treatment plants are critical hotspots almost anywhere. And why does, it clearly, right? It all concentrates from every single person into those particular spots. And so the question becomes, what do we do with that effluent? What do we do with that waste, right? And this was seen, for instance, where in another study by others where there was waste that was being used, I think maybe from Boston or in some particular place. And waste comes into most of these wastewater treatment plants, and there's a settling process. All the solid waste kind of settles to the bottom before there's treatment of the liquid waste. And you kind of make these kind of, cakes, if you will, kind of euphemistic word for the material that remains in the wastewater treatment system. And then those are used as fertilizer in various places. That's great because it's really high biological material, but nothing is done to the DNA. And so at some point, someone said, well, where are those cakes being used? They were being used to fertilize a lot of the citrus fields in Florida. And so then they went and looked at the oranges in those particular fields, and lo and behold, they had resistance genes and their bugs on them that came originally sourced from human fecal samples. So that's the identification of a hotspot. And I think about, okay, well, maybe that's not such a wonderful idea. Maybe you need techniques to basically degrade that DNA as well. So I think that's the status of hotspot detection. I'm not sure that there are truly cold spots, except for maybe to say, by evaluating different decontamination protocols, we can kind of mitigate hotspots. Maybe that's the easiest way to think about this. Right.
Thank you.
Alex, you had a question.
Yeah, okay. So unlike many other countries, in India, there are little to no regulations on buying antibiotics. So, but if this was stopped, a lot of livelihoods of people, farmers, and anyone who really depends on it would be affected, right? So this is kind of a TOK question, not really related to the first half, but since you're so involved in research, what do you think? How can research have an adverse effect on people's livelihood? Like the
That's a great question. And you know what I'm going to do is hopefully any good TA is start to do is maybe turn that question on its head. And that is to say, why do we think of public health measures that are clearly going to have some benefit as necessarily only encoding costs, right? So the way the question was posed was, right, well, we could try to mitigate antimicrobial production or use, but maybe that's going to hurt livelihoods. Now, all of this, if you want to use economic arguments, needs to consider some cost benefit analysis. So what is the cost of allowing a country like India to become a hotspot for antimicrobial resistance dissemination, right? What is the cost, even though it's long-term, of the risk that you're buying yourself, right, for having infections that are going to become resistant at a faster clip than anywhere else? And that needs to be weighed against the idea that, okay, right, maybe new economic incentive structures need to be put in to protect those folks whose livelihoods depend on, for instance, the production of antimicrobials, right? So it is true, India is the largest producer in the world of generic antimicrobials, right? And there were actually, I think one of the scariest studies I read was actually right as I was transitioning from being a postdoc over to being a faculty member, so 12 or 13 years ago. And there was a Swedish group that had done some work in a couple of villages in India, and I can't quite remember what part of India this was, but it was where there were some large generic manufacturers of antimicrobials, right? And so they had the hypothesis that if these particular factories that were producing antimicrobials at large scales, if they were not being really good about disposing waste and dealing with sort of potential contamination, that they might see enrichment for antimicrobial resistance genes geographically close to those particular factories, right? And but what they found was sort of mind-blowing. It was way worse than what you expect, right? So as it turns out, not only was this particular group of factories not very good with their contamination, they were just dumping their particular waste directly into the water stream system directly next to that factory. To what extent were they doing this? When this group went in and they sampled the water from that particular river that was next to the factory, the concentration of antibiotics in that water was equal to the concentration that you would take from a pill, right? So that is to say, you could just drink that water and you would get a good cocktail of antibiotics. You wouldn't even have to buy the pill, right? So just think about what that means. That means the concentration of that waste at the source was so high that even after it got diluted in the wastewater system, it was exceptionally high. They then went a couple villages down into their water system, and they had to go 2 or 3 shells of villages out when they looked at their well water, and they could still find detectable levels of antibiotics, right? Now, I would argue that you're not hurting anyone's livelihood if you were to just stop a practice of that type, right? You could still make all of those generic antimicrobials, and India should incentivize, its workforce to produce, anything that is of particular value that could be sold on the global market. But in doing so, right, that example I gave you is hurting Indians, right? It is first the local problem. So I think there are balances that can be put into place to say, this is not advocating for not making antibiotics, right? It's advocating for the appropriate regulation. And I think one of the most successful agencies, I think it's called CDDEP. It doesn't roll off your tongue, but it's an organization I think originally started in DC, but run by a guy, at least of Indian origin, who now I think lives in Delhi. His name is, I think, Ramanan Lakshmi Narayan. And it's a great network of folks looking at infectious diseases. And he basically made the decision, and him and his colleagues, to say, if we want to do stuff about antimicrobial resistance and antibiotics, we have to pitch this in terms of economic terms, right? We need to convert all of this cost in terms of human lives and drug resistance into dollars and rupees, right? And only then will we get across to the economic minister, who's going to just think from election cycle to election cycle and say, look, you need to think about the fact that the amount of money used to treat people, the amount of money used to have unmitigated epidemic or even pandemic level disasters with drug resistance is ultimately bottom line dollars, right? And then from that perspective, you might be able to say, this is the reason why you have a collaboration between regulation and the people making whatever they're making, to say, isn't ultimately your best interest to treat this particular problem. It's not an easy deal, because the problem is, I think most of us as humans are just sort of, the way our society works on local short timescales. To convince people to think in longer term timescales is much harder, right? You're like, oh, that's someone else's problem. Why do we have an unmitigated, climate health disaster? Because look, not going to impact me, it's going to impact my grandkids. So why don't I guzzle gas for a little while, right? Same mentality. And so I think it is this thing to convert some of those economic challenges into local challenges. Like, you know what? Think about how much money is wasted when people are in a hospital or succumbing to an infection because of drug-resistant infections, right? Think of the global hit or the huge economic hit to the Indian economy when that happens, right? And so for that, if you were to be a little bit better with being stewards of antimicrobials, right, you can help with that. Another thing is, basically, if you keep abusing your antimicrobials, they will not be able to be used anymore, and you're cutting yourself off from future revenue because you're making your drug less effective because drug resistance is going to go up. So, again, you asked for a TOK answer, and that's my attempt at one.
Okay, are you okay with one more question from Shothiba, and then we'll wrap
Sure.
up? Thank you so much for your patience.
Oh, no problem.
So my question is regarding your life at KIS. So what did you do for your biology IEA?
My biology, independent study? Is that what you were asking?
I think so that's what it was called then, yeah. Or did you do an extended essay in biology?
Yes, I did an extended essay. So actually, it was I worked under Mr. Nanan's guidance to look. I worked with a local PhD student who was trying to assess the allergenic potential of pollen from plants in the Palni Hills. And so my extended essay was essentially on sort of piggybacking on his research, doing some protein biochemistry assays to essentially understand what the protein content was of those allergens. So that was.
Great, thank you, Gautam. That was very informative, and I liked the way we ended back in Cody. I'm actually going to piggyback on someone's words in the chat box here. It's from Viva from class of 1956. She says, a terrific presentation, a discussion of such an urgent situation. Your work and the way you go about it is amazing, and you're an inspiration to the current generation of Cody students. And I have to say, so true, and you are a true inspiration for us. And thank you for keeping us so proud of your work and your group, what you do at the lab. And thank you for indulging us also with your time.
It's been my pleasure, Manju.
I know I haven't even gone through all the questions in the Q&A box still. And it's just, brimming where you've opened our perspective there. Thank you so much. Real pleasure of having you here.
Absolutely. Thank you very much for doing this. I think the quad series is great. And again, hopefully at some point this will happen in person and you can come and visit.
Yes, so hopefully we can have you in person here and have the family up here as well.
Absolutely.
All right.
All right.
Take
well,
care.
thank you all.
Good night. Yeah. Have a great weekend.
Good afternoon. Good night for all of you.
All right. Good night.
Bye.
Bye.